Again, welcome everyone, and thank you for joining today's presentation. Just a reminder to please keep your microphones on mute and to follow the participation link if you are looking for that participation certificate. Okay, so today we'll be taking a look at the Illumina MiSeq. If you remember from last week, if you joined those presentations, we heard about the iSeq from Maria and the MiniSeq from John. In the next presentation at 11, if you join that, John will be taking you through the NextSeq.
Hi Jane, sorry, it's John. Just wanted to let you know you've put yourself on mute again, so we can't hear you.
Oh dear, sorry about that. Not off to a great start. Okay, welcome everyone, and thank you again for joining. Holly, I think you can go ahead and start the recording. If you're looking for the participation link, please don't forget to follow that link if you are looking for that certificate. If I can just remind everyone to please keep their microphones on mute, and we'll have a section at the end of the presentation for questions. So just to look at today's agenda, we're going to run through an introduction to the MiSeq. If you were on last week's presentations, you would have heard about the iSeq from Maria and the MiniSeq from John. In the next presentation at 11, John will be talking about the NextSeq. So for today's presentation, we're just going to be looking at an introduction to the MiSeq, the benefits of the system, a quick look at the hardware, and then we're going to be focusing on some key applications. And these applications will involve specifically to do with library prep using the Nextera Flex library prep kit. And then we will have some time for questions. So please feel free to write your questions in the chat so that we can come back to them during the question section. Okay, just an introduction to the MiSeq. The MiSeq is actually Illumina's oldest benchtop system. It was launched in 2011, but it's still the most popular of Illumina's sequencing systems. We have over 6,000 MiSeqs worldwide, and we have about 21 MiSeqs in South Africa. It's extremely popular. First of all, because it is the system with the longest reads available from Illumina. We'll take a look at that just now. It's also extremely popular because of its flexibility and scalability. And so it can cover really a wide range of applications in different fields, from oncology to microbiology and complex diseases. It has fast turnaround time and proven chemistry. And it's really the workhorse of the sequencing industry, and it's known to be extremely reliable. So just to highlight the benefits there's just the main one being that it provides the longest reads, up to 600 bases. So for paired-end sequencing, that's 300 times 2. We'll take a look now at how you can achieve that. And these long reads really open you up to new applications, making the platform ideal for applications like metagenomics or de novo sequencing, where you really require longer read lengths. Okay, so if we just take a quick look at the system specifications, when you are looking at a system, there's two main specifications that you're looking at, and that's the quality score, the Q30 score, and the amount of output that you can achieve. So for the MiSeq, you get in a quality score of over a Q30 score of over 75% for paired end 300 cycle reads. And and over 85% for paired end 75 cycle reads. You get up to from 0.3 to 15 gigabases of output data. So we take a look at that now as to why the system is so flexible, and from 1 to 25 million reads or clusters on your flow cell. So this scalability really has to do with the number and configuration of consumable kits available. So if we just take a look at that, you have your version 2 consumables, which are your standard consumables, and they are available in both nano and micro configurations as well. And then you have your standard version 2 chemistry, giving you up to 15 million reads. We then have your version 3 chemistry, which is the improved chemistry. This just gives you higher cluster density, so you have more clusters on your flow cell, and it also allows for longer read length and improved quality scores. So that's with your version 3 chemistry, where you can get 600 cycles. But you have your nano and micro configurations too on the version 2 chemistry. So this just shows you how you can get how the MiSeq supports a broad range of applications, because you can pick the perfect number of cycles to meet your needs and the amount of output data that you need to meet your application. If we take a look at where the MiSeq fits in to Illumina's sequencing portfolio, it's obviously one of the benchtop sequences, and it fits nicely within the middle. Just get my pointer. So you heard last week about the iSeq and the MiniSeq, so obviously we're going in order, and the MiSeq fits quite nicely. You get 25 million reads, which is the same as the MiniSeq, but you'll see they double the output data. You'll see quite a big jump between the MiSeq and the NextSeq, which John will talk about later. Your NextSeq, the flow cell has 4 lanes instead of 1, so you get in a lot more data with the NextSeq. So it really depends on the application that you're interested in and the coverage that you require, and obviously the throughput that you have. Because as you go scale up with the instruments, you're going to get a decrease in price per gigabase. But obviously it would also depend on the throughput that you have as to whether you can support one of these large production scale sequences. Okay, so just a quick look at the MiSeq hardware. As we said, it's one of the older systems, your oldest benchtop system. So it still has the reagent cartridge chiller to keep the reagents at 4 degrees. And you'll see some differences between the system and the newer systems, where they are now using buffer cartridges and waste cartridges instead of bottles. And you'll see some improvements with the newer systems in terms of the optics and the fluidics. But with your MiSeq, all your reagent consumables are RFID encoded, which also just allows you to track your consumables. Okay, so just something to remember, and I think you will remember from previous talks if you joined in on those, is that the MiSeq uses what we call a 4-channel SPS chemistry. So as it's one of the earlier sequences, along with the HiSeq, the MiSeq is still using this 4-channel chemistry, whereas your newer systems, you would have heard last week, your iSeq is using your 1-channel chemistry, and your MiniSeq was using your 2-channel chemistry. So the 4-channel chemistry, that just means that each of your 4 bases are fluorescently labeled, and they're and it therefore means that 4 separate images need to be taken by your camera and analyzed, which means a slightly longer image in time. But for the MiSeq, if you're working with really low diversity libraries, if you amplicon sequencing, 16S RRNA sequencing, it's the 4-channel chemistry is actually better suited. Okay, so just to take a closer look at that sequencing by synthesis, what's happening there as during each cycle as a basis is being incorporated. Remember, each one has its own individual fluorescent label. That base is then being excited by your light source, and then each one is emitting a different wavelength, and you have the images being captured. So you're having 4 images that are being taken during every cycle. Okay, so a quick comparison between the 4-channel chemistry versus the 2 and the 1-channel chemistry. Your 2-channel chemistry is sort of considered then the next generation in chemistry, because it's obviously quicker because you only have 2 images that are being taken and analyzed. So here you only have 2 fluorescent labels. Your T's are, say for example, fluorescently labeled with the green dye, and your C's red, and your G's are not. There's no fluorescent labeled with your G's, and your A's is half of them are green and half of them are red. So in this way, your system can take 2 images and analyze as to which base was incorporated into that cluster. For your 1-channel chemistry, you're only making use of 1 fluorescent dye, and you'll see that your iSeq is making use of this, taking advantage of this chemistry. And you would have seen in last week's presentation that your iSeq actually has that optics built into the cartridge itself, because you're only looking for 1 fluorescent dye. You're almost sort of looking at flashes, and you have that logic between sort of an on-off signal between the two images to work out the which base was incorporated into that cluster for each cluster. Okay, so as we said, the MiSeq is still using the 4-channel chemistry. If we take a look at the MiSeq's application portfolio, the MiSeq supports several key applications, from small whole genome sequencing to targeted gene sequencing, amplicon sequencing, to 16 RRNA sequencing, de novo sequencing, and many more applications. Actually, your small whole genome sequencing and your 16S RRNA sequencing are very popular on the MiSeq. And the reason you have able to support this range of applications is to remember all those different chemistry options that are available. Okay, so if you look at these key applications, it actually makes the MiSeq the perfect system for the field of microbiology, from small whole genome sequencing for microbial identification. This helps obviously researchers gain genetic insights into bacteria and viruses, and also to identify drug relationships. To shotgun metagenomics, John will be taking a look at that in the next presentation in a bit more detail. To 16S RRNA sequencing, which helps researchers clarify phylogenetic relationships between bacteria and fungi. To virology, we're looking at the detection of low frequency viral particles within non-cultured samples. So when it comes to library prep, basically all your library prep workflows obviously have a common workflow, which is obviously first your sample prep to do your extraction of your RNA or DNA. Moving on to using the library prep kit specific to your application, which all have the same common goal, which is to add in those special adapter sequences that will ultimately have oligo regions that will bind to, that are complementary to the oligos on your flow cell, and also include the sequencing primers and obviously your index. And then the next step is to sequence on your instrument, in this case the MiSeq, and then moving on to data analysis. So all library prep sort of follow that same workflow. If we look at the different library prep kits that are available for the MiSeq, last week John took you through the small RNA, and later we'll talk a bit more about RNA-seq. Maria went through the AmpliSeq for Illumina last week. Today we are going to actually focus on Nextera DNA Flex. So it's actually only fitting, if we're going to look at one specific library prep kit, it's only fitting that we look at Nextera DNA Flex, because just like the MiSeq, it's extremely versatile and flexible. So it's a great kit to take a look at with this presentation. Just to note something, Nextera DNA Flex is actually now being renamed Illumina DNA Prep, but for the purpose of this presentation, we're still going to refer to it by the name that most people will know, which is Nextera DNA Flex. But just to note, if you're looking for it, it is now being renamed Illumina DNA Prep. So Nextera DNA Flex uses a new innovative technology, which you'll know already from Nextera kits that involve this tagmentation process using transposons. With DNA Flex, these transposons are linked to a magnetic bead. And this just means that when your tagmentation is taking place, which is to both tag and fragment your DNA, this is happening in a very uniform and reproducible manner because of these transposons that are linked in a uniform manner to your bead. If we take a look at the workflow just to help understand, what happens is you obviously first isolate your genomic DNA, and then you add in your beads to your DNA input. Here the kit can support a wide range of DNA concentrations from 1 to 500 nanograms, but what's important to note is that if you really want to take advantage of the benefits of this technology, is if using a concentration between 100 and 500 nanograms, you actually can then achieve normalization in one step. Because what's important to note is that when tagmentation is occurring, this DNA actually remains bound to the bead. So it's once you've saturated the beads, no more tagmentation can occur. And this then results in a normalized library if you are working between 100 and 500 nanograms. So you also get consistent insert size just because of the uniformity of those transposons on the bead surface. Okay, so following that tagmentation step, the next step is adding in the adapters, and that is done through PCR. So these tags that were included by the transposons are complementary to your read1 and read2 sequencing primers of your adapters. And excuse me. And then through PCR, you have incorporation of your adapters on either side of your fragment. Okay, so your now normalized library is ready to sequence. They do say that okay, QC and normalization are then not required. We obviously would always recommend that you still do your QC step afterwards, just to see if you have any artifacts from PCR, possibly adapted diamonds. So we wouldn't say skip that step, but you should have a very consistent normalized library if you were working within this concentration range. So there's no need to really QC your DNA up front. If you know it's within this range, you don't need an exact concentration because you will, with this input, you will have a normalized library. Okay, so with Nextera Flex, you really are able to save time and increase efficiency compared to other library prep kits. You can actually do the entire library prep workflow in around 3 hours, which is great. It obviously is integrating a few of the steps into one easy to use workflow. You can also work, and you can take a look at this. You can actually work from saliva or blood samples or dried blood spots, and you can buy an extra reagent there to do the lysis from those samples, and that is a very quick protocol that can occur within half an hour. So if you're working from those samples, you can go from extraction all the way through to library prep within 3.5 hours. Okay, so Flex covers a whole range of applications, and we're going to take a look at some of these in a bit more detail, and specifically those that are perfect to fit with the MiSeq. But this range of applications, from whole genome sequencing to de novo sequencing, shotgun metagenomics, your 16S RRNA, viral sequencing, and etc. Mitochondrial DNA sequencing, we'll also take a look at. Okay, so when looking at whole genome sequencing, your Nextera Flex has the flexibility to cover across small, large to human complex genomes. But obviously on the MiSeq, we'll be looking at small whole genome sequencing. In the next presentation, Jean will be focusing on a bit more with the Nextseq, that you might be looking at applications that involve large whole genome sequencing. Okay, so next, just to take a look at 16S ribosomal RRNA sequencing. It's a common amplicon sequencing method used to identify and compare bacteria or fungi present within a given sample. So it's just a well-established method that allows the phylogeny and taxonomy of samples from complex microbiomes or from environments that are difficult to study. Here with Nextera Flex, you can specifically look at the V3 and V4 hypervariable regions. And this workflow can also be adapted to look at your ITS regions if you're working with fungal samples. So moving on from, if you are looking at specifically environmental metagenomics or eDNA, as they call it, then Nextera Flex is also great for that. Obviously, if you're working with the 16S RRNA sequencing or different approaches, shotgun metagenomics, or whole genome sequencing. Here it's NGS has become a great way to investigate new methods for understanding environmental influences, especially when it comes to water sources and water quality. Or other examples include profiling of microbial populations from samples taken from ports or deep sea ocean vents, for example, or agricultural samples, soil samples, for example, to understand the microbial profile in those samples. So this really promises to be a vital tool for biomonitoring and conservation. Again, NGS can also be used to explore the human microbiome, and again, the great application for Nextera Flex and the MiSeq. NGS has led to the emergence of new therapeutic approaches, either by removing harmful taxa or reinstating missing beneficial taxa, and understanding the functional roles that they perform. For example, your human microbiome can affect the human immune system, skin disorders, inflammatory responses, as well as metabolic and neurological disorders. So it really can help you understand, for example, a relationship of the gut microbiome to neurological disorders such as autism, yeah, this interaction between human and microbiomes can just also play a key role in determining the differences between a healthy state and a disease state within the human body. So, okay, and then moving on to another key application that's great with Nextera Flex is when it comes to viral sequencing. If you're working with RNA viruses, obviously you would first need to perform cDNA synthesis to convert your your RNA to double-stranded cDNA, which is the input for this sample prep workflow. You would then make use of Nextera Flex for whole gene, if you're interested in whole genome sequencing, or possibly you're interested in enrichment with specific viral targeting panels. So, for example, we have the respiratory virus oligo panel, which is an enrichment-based protocol. So here you're using Nextera Flex, but following with an enrichment step. So within this enrichment step, which we'll take a look at just now, your specific probes for this panel are bound to biotin, and after preparing your libraries, you can then pool these samples together and hybridize your biotinylated probes for enrichment. With this specific oligo panel, you can look at 30 different respiratory viruses. So, for example, included in this panel is influenza, the different human adenoviruses, and the different coronaviruses, to name just a few. And then just to highlight how this enrichment works, once you've pooled your libraries, you can then go through a hybridization step to hybridize these biotinylated probes, and then using your strict dividend beads, you can isolate those fragments, and then you would wash off the unhybridized fragments, so you get enrichment of the fragments that you're interested in. Okay, next to just take a look at mitochondrial DNA sequencing. It's a useful tool for researchers to study in human diseases such as diabetes and certain cancers, and as well as mechanisms of aging. So here you would use Nextera Flex for either a targeted sequencing approach or whole exome sequencing, which would provide a complete picture of the coding regions of the mitochondrial genome, or if you're looking at a targeted sequencing focusing on particular genes or regions. This sort of really, if you have a prior knowledge of specific diseases and their associated variants. And then lastly, I just wanted to focus on plasmid sequencing that can also be done with NGS using Nextera Flex, and on the MiSeq, if you will obviously know of plasmid sequencing can be done with Sanger sequencing, but if you're really looking for a sort of to provide you with the complete plasmid sequence, as well as if you're working with a large number of constructs, then NGS has its advantages. You get validation of the known plasmids, and you can really sequence the full length of the plasmid. If your plasmids are unknown, you don't need any reference sequences required either. So this really just lowers your cost of sequencing, especially if you're working with a large number of construct variants. So I'd like to also just end off by highlighting the advantages of NGS versus your traditional methods. Your traditional methods, if you're looking at PCR-based screening or Sanger sequencing or even capillary electrophoresis, they've been around since the 80s and 90s, but ultimately the disadvantages there is that they lack sensitivity. You also require a prior knowledge of your targets, and you're working with low sample throughput. Whereas with the advantages, obviously, of NGS, is much more sensitive, and you can multiplex multiple samples, and you really have a lot of advantages when it comes to NGS. And again, I mean, where your qPCR and Sanger technologies are inherently flawed is when it comes to scaling up projects. So with NGS, you can really scale up and enhance your detection power. And it's also much more cost effective when it comes to scaling up compared to other technologies. Okay, so I would just like to summarize when it comes to the MiSeq and what we've gone over through today. If you remember, things to keep in mind, that the MiSeq is obviously extremely flexible benchtop sequencing system. The number of kits, reagent kits and configurations available make it extremely scalable to meet your needs. It's extremely versatile, and it can cover a range of applications, and it really is a great partner with Nextera DNA Flex, amongst obviously all the other library prep kits that are available for the MiSeq. That is all that I have for today. Do we have any questions? Maybe Holly, sorry, if you can just check in the chat. I don't have that screen open.
There don't seem to be any questions.
Okay, just give a little moment if anyone has any questions, that they can just post them in the chat, or you can obviously unmute yourself. We have ended a little bit early, so if you would maybe take a short break, and then at around at 11, John will be introducing us to the Nextseq.
Jane, thank you so much. Thanks so much for the awesome presentation. It's always so nice to hear about the MiSeq. It's a very flexible, very versatile instrument. So it's always so nice to hear about some of the applications that you can do on the MiSeq. I'm sure everybody always has a soft spot for the MiSeq because it's such a lovely instrument.
Yes, indeed. Okay, great. So no questions. All right, then we'll be seeing you all back at 11. Thank you very much, everyone, for joining, and please don't forget to follow that participation link if you're looking for that certificate.
Perfect. Thanks so much.